Morning, students. Today I am going to resume teaching after a long gap. I will deal with the immunopathology which has been allotted to me. And in this, first of all, I would like to tell you the broad topics which I am going to cover. That is, definitions of common terms used in this chapter, normal immunity, hypersensitivity states, autoimmune diseases, immunodeficiency diseases, and amyloidosis. These are the broad chapters. Now, in the definition, what is immunology? It is study of structure and function of immune system. What is immunity? It is protection of body from infectious microbial pathogens. It may be innate or adaptive. It may be active or passive. It may be humoral or cell-mediated. Now, what is immune response? It is a collective and coordinated induction of protective host reactions against microbes, tumors, their products, and dead cells. What is the immune system? Immune system is constituted by molecules, cells, tissues, and organs which protect our body from pathogenic microbes present in the environment. Now, what is immunodeficiency? It is failure of immune system, and it may be of two types, primary or inherited, secondary or acquired. What is hypersensitivity or allergy? It is exaggerated immune response to an antigen producing tissue injury. What is immunological tolerance? It is lack of responsiveness by immune system to an antigen exposed to our tissues. What is self-tolerance? Self-tolerance is lack of responsiveness by immune system to body's own antigens. What is autoimmunity? Autoimmunity is immune response against self-antigen, and it is produced by when the self-tolerance is broken down due to some reason. What is the amyloidosis? It is a disease with deposition in tissues of abnormal proteins, some derived from immunoglobulin fragments. Now, I will discuss in detail about the immune system. The immune system, it is constituted by tissues, cells, and molecules that you have already seen. So what are the tissues or organs of the immune system? These may be classified into two types. One is generative or primary or central. Others are the peripheral or secondary organs. The central organ, primary organ, are the thymus, where the T lymphocytes develop, and bone marrow, where the blood cells are produced and B lymphocytes mature. While the peripheral organs are spleen, lymph nodes, and the lymphoid tissues. So, spleen is an abnormal organ to trap blood-borne antigens. Lymph nodes are nodular aggregates of lymphoid tissues along lymphatic channels throughout body, where microbial antigens entering through epithelia and colonizing in tissue are brought by antigen presenting cells. Means to say that once the infection microbes enter the body by breach of the epithelium, they enter into the cells, they colonize the tissue, and from there they are trapped by the antigen presenting cells. Antigen presenting cells bring those microbes through the lymphatic channels to the lymph nodes. Now, the third category of the peripheral immune organs are cutaneous lymphoid tissue, and fourth is the mucosal lymphoid tissue. Both of these lie under the epithelia. Now, the types of the mucosal lymphoid tissues are, it may be in the respiratory tract, it may be in the gastrointestinal tract. So, in the gastrointestinal tract, you know the major lymphoid organs are the pharyngeal adenoids and tonsils, pairs, patches of the intestine, and appendix. These are the lymphoid organs present in the epithelium of the gastrointestinal tract. Now, you can see one thing here is that the lymphoid organs which are present beneath the mucosal cells, mucosa, at any time the body contains more than half of the lymphocytes of the body in this area, that is beneath the mucous membrane, and most of them are memory cells. Now we will proceed to the cells of the immune system. What are they? They may be, number A, they may be cells of innate immunity or primary immunity, and cells of the adaptive immunity. The cells of innate immunity are natural killer cells, that is NK cells, and the innate lymphoid cells, that is ILCs. These are the cells of innate immunity. And cells of the adaptive immunity are of different types. Number one is the granulocytes. Now granulocytes, you already know, are the blood cells which cytoplasm contains certain granules. That's why these are known as granulocytes. This may be of three types: neutrophils, eosinophils, and basophils. The major difference between these two granulocytes are that the neutrophils and eosinophils contain fine granules, while the basophils contain very coarse granules. The color, the staining of the fine granule in the neutrophil is deep purple, while staining of the granules of the eosinophil is red or orange. Besides that, the characteristic of nucleus, what is that? The nucleus of the neutrophil is multilobed, while the nucleus of the eosinophil is bilobed or kidney-shaped, and the nucleus of the basophil is not usually visible due to the deep staining, very coarse, deep purple staining granules which cover the nucleus. So these are the characteristics of the granulocytes. Now come to the lymphocytes. Lymphocytes have been classified differently according to size. It may be said the small lymphocytes and large lymphocytes. In the small lymphocytes, there is very minimal perinuclear cytoplasm, while in the large lymphocytes, the amount of cytoplasm around the nucleus, single nucleus, is more. Second classification is according to the stage of the activation of the lymphocytes. According to that, it may be naive lymphocyte, which is immunologically inefficient. It may be effector lymphocytes, which is mature enough to eliminate microbes, and it may be memory lymphocytes, which are aware enough to rapidly kill microbes if they return into the body. So these are different types of the lymphocytes according to the stage of activation. Now the types of the lymphocytes according to the function. They may be B lymphocytes, which differentiate into plasma cells which produce antibodies and against the extracellular microbial antigens, and this is known as the humoral immunity. The T lymphocytes, it may be of different types. They, according to the function, number one is the helper or CD4 plus T cells. These T cells, helper CD4 helper T cells, produce cytokines which aid in differentiation of the B lymphocytes into the plasma cell, as you have seen in the case of the humoral immunity. Another, the cytokines are produced to activate the macrophages to phagocytose the microbes. So these are the function of the helper T cells. And the second category is the cell-mediated immunity, while the previous one is the humoral immunity. Now, second type, functional type of the T lymphocyte is the cytotoxic CD8 positive T lymphocytes, also known as CTC, cytotoxic T lymphocyte. It kills the antigen presenting cells infected with microbes. Next is the regulatory T lymphocytes. Its function is to suppress exaggerated immune response. Now come to another category of the cells of the adaptive immunity, and that is the antigen presenting cells, also known as APCs. These present antigens to T cells after capturing microbes that enter body after epithelial breach, as you have seen earlier. Now, what are different types of the antigen presenting cells? Number one is the dendritic histiocytes, which are present in tissues and beneath the epithelia. Second is the lymph node dendritic cells. It may be follicular dendritic cells which activate B lymphocytes, and paracortical dendritic cells present antigens to the T cells. Now, third category is the epidermal or mucosal Langerhans cells which are present in oral, anal and vaginal cavities. Everywhere it is there. Next category of the cells which form the immune system is the macrophages. Actually, macrophages may be first classified into it is the part of system which is known as monocyte macrophage system or reticuloendothelial system, MMS or RES. Now it may be broadly again classified into two types. One is the circulating macrophage, that is known as monocytes, about which you already know, you see that in the blood film, and another is the tissue macrophages. There is a big list of tissue macrophages about which you must be aware, and we are going to deal with it. Number one tissue macrophage is the reticulum cells, and it is present in the bone marrow. Second one is the osteoclast, present in bone. Histiocytes, present in connective tissue. Kupffer cells, present in liver. Littoral cells, present in spleen. This is also known as sinus histiocytes. Next one is the mesangial cells. Mesangial cells which is present in the renal glomerulus. Hofbauer cells which is present in the placenta. The Langerhans cells which is present in the epidermis and mucosa. Dendritic histiocytes which is present in the skin, and microglia which is present in the CNS, and alveolar macrophages which is present in the lungs. In that way you can see the macrophage is present all over the body in different systems to cause the phagocytosis of the pathogens. Now after that we shall deal with the molecules of the immune system, and first we shall deal with the molecules of innate immunity. These are number one adhesion molecules, defensins, and third one is the complements of alternative and lectin pathways. These are three molecules of the innate system. The fourth one is the pattern recognition receptor molecules, and these may be of three types. In the plasma membrane receptor is for the external microbes, the endosomal receptors are for the ingested microbes, and the cytosolic receptors are for the cytoplasmic microbes. Now the fifth category of the molecule is the interleukin 1 and interferon 1 and gamma. These all cells are the molecules of the innate immunity. Now come to the molecules of the adaptive immunity. These are the complements of the classical pathway. These are the molecules secreted by the helper T cells, that is cytokines. These cytokines are secreted by the innate immunity also, as you have seen the interleukins, and these are secreted by the helper T cell, that is adaptive immunity also. Next is the major histocompatibility complex, that is Msc molecules. Msc molecules are of two types, Msc1 and Msc2. The Msc1 molecule is present in the innate immunity also and adaptive immunity also. That is the sixth category of molecule in the innate immunity. Msc1 molecule is present on the surface of body cells, all cells of the body, and they bind to inhibitory receptor of NK cells and virus. So and virus infecting the cell inhibits Msc1 molecules expression, so that NK cells is no more inhibited, destroying the virus infected cell. So means to say that so far there is no virus inside the cell. The NK cell which destroys the any cell is remain inhibited. Once the virus infects it, the inhibitory receptor is no more active, and the activating receptors become active, and that causes the destruction of the cell. which is present, which destroys the cell inside which the virus is present. So virus is the stimulating factor for destruction by the NK cell. Now come to the Msc molecule of the innate adaptive immunity. The MSC1 molecule of the adaptive immunity are present on the virus infected antigen presenting cell, and they linked to the T cell receptor of CD8 plus T cells. That is unique. It is similar to the MSC1 molecule of the innate immunity. And another MSC2 molecule is present on the antigen presenting cell membrane, and it links to the T cell receptor of CD4 T helper cells, and by that the CD4 T helper cells releases the cytokines, which on one hand activates the macrophages to produce the causes the on one hand they activate the B lymphocytes to be converted into plasma cell to produce antibody, and on the other hand they activate the macrophages to activate it for the phagocytosis. That's what all about today, and in next lecture we shall discuss the further subtopics. Okay.